Yeah. So, hello everyone. For those I have not met yet, I am Kevin Knudsen, president of the ABRF, and it is my privilege and pleasure to welcome everyone to the 34th annual ABRF meeting. This is the first time we've had the ABRF annual meeting in Boston, and we are really excited to be here, as this is a city rich in American history and achievement, and I think we'd all agree it's one of the world's leading areas for biotechnology, which is at the core of our society, pun intended. But we do acknowledge that this land is the territory of the Massachusetts, Pawtucket, and their neighbors, the Wampanoag and Nipmuc peoples, who have stewarded this land for hundreds of generations. We recognize the repeated violations of sovereignty, territory, and water perpetrated by invaders who have impacted the original inhabitants for this land for over 400 years. As visitors, we extend our respect to the citizens of these nations who live here today, their ancestors who have lived here for over 500 generations, and to all the indigenous people. We also affirm that this acknowledgement is insufficient and does not undo the harm that has been done against indigenous people in their land and their water. With that said, we are looking forward to honoring and representing these indigenous people by being good stewards of our meeting. As visitors, we extend, and as I look at the program, I'm excited by its content, the breadth of sessions that I'm sure will have many topics of interest to those who work in and support CORES. This meeting has achieved a monumental benchmark. We have exceeded and surpassed 1,000 registrants. I believe well done. I think the official number that I heard was 1,049 from about an hour ago. Congratulations to all those who worked so hard to put this program together and bring it to us. This was certainly a team effort, and I'd like to thank the program committee, chaired by Andrew Vennard, AMR, and for all they have done to bring this program to me. When you see them, thank them. They've put in tireless hours to make this happen. I'd also like to thank our institutional and group members and corporate partners for your support in this meeting and our organization. Thanks to all our members and to all you do and for coming to this meeting and joining us today. I look forward to talking science, sharing ideas around core operations with all of you in the upcoming days. And without further ado, I'd like to hand this meeting off to our program chair, Andrew Vennard.
Thank you, Kevin, and welcome again, everyone, to Boston. I'm incredibly excited. Sorry, I'm losing my voice, so people probably are not used to me not talking, but I'll do my best here. I'm incredibly excited for our program this year, and we'll tell you a bit more about that shortly. But first, I'd like to share some gratitude. The theme this year is driving collaborative science, which is at the heart of what CORES do, and this is the case with organizing this meeting. While I had the pleasure of leading, the true generators of the program include Marie Adams, Jim Chambers, who also has the pleasure of having to work with me on a day-to-day basis. Sorry, Jim. Kathy Cormier, Caroline Deneff, Chris Gilpin, Tricia Rogers, Bob Steen, Sue Weintraub, and Stuart Levine, who I hope all 1,000 plus of you take time to thank, as he had a significant part in filling many of these sessions after planning a full meeting and seeing its conversion to virtual just 2 years ago. Thank you all. To the AMR team, especially Lindsay Plath, Hunter Stoll, and Leslie Kowalski, who worked with us throughout the planning process with ample patience for my constant delays and are here making sure everything goes smoothly. Thank you. And of course to Ken Shopman, ABRF's executive director, who keeps us all in line. Thank you. Last but certainly not least, thank you to every individual session organizer, to our speakers, our contributors. Without you, none of the great conversations, education, and learning to happen over the next few days would be possible. The program will feel familiar to many, but has slightly different format this year. We have 3 plenary blocks with 2 sessions per block over the next 2 days. The intent is to hear multiple perspectives on a similar theme. These will be followed by traditional breakouts, many of which will be continuations of the conversations from the plenaries. We then hope you have a hard time deciding what session to attend during the remaining current breakout sessions. While some of the sessions will be organized purely on a technology track, many others are meant to provide resources depending on where you are in your specific career path. Please remember, if you do have a hard time choosing what to attend, recording of the program sessions will be available on the meeting website the day after they are presented. On Wednesday, we will start with our DEI award, and then we're going to have an emphasis on the rest of the day on community colleges and how we as core facilities can work together. We've extended an invitation to many local community colleges and are excited to have a lot of folks join us. Please make sure you make time to engage with these folks and let them know what our world is about and see how we can make sure we bring them more into ours. We've built buffer time at the end of each of our sessions for breaks and refreshment and primarily engagement with our vendors. Please remember to visit the ABRF exhibit hall and engage with our sponsors who make this event possible. The event hall will be open Monday and Tuesday from 9 to 4:30 and on Wednesday from 10 to 4. Our poster sessions will occur during lunch breaks this year, which we've extended to 2 hours every day. The 2023 technology showcases feature our leading partners who will review the latest instrumentation and software solutions. These showcases take place on Monday and Tuesday from 7:30 to 8:30 in the morning and 6 to 7 in the evening, and again on Wednesday from 7:30 to 8 in the morning. And of course, our closing social will be Wednesday in the Great Green Walls of Fenway Park. We hope to see you all there. A few more additional housekeeping items. Please make sure to silence your phones. Photography and video recording of sessions are prohibited. However, documentation of the meeting as a whole is something we love. ABRF's social media policy is that everything can be shared. And if you do post, the hashtag for this year is ABRF 2023. If a speaker asks that their presentation not be shared, please respect and do not post that on any media platforms. For our remote participants, thank you for joining us as well. You may post questions in the online chat. If you're here in person, please make sure you use the microphone so that everybody can hear and the recording can pick it up. Last and most importantly, I talk fast, I mumble, and the voice is just about gone. So all of this information can be found in the meeting app. Please be sure to download it so you can access all poster and session abstracts, connect with each other, and create your own personalized meeting schedule. With that, I'm sure I've probably went over my time, so I'll invite Dr. Jackson Buss, research scientist with NEB, our Platinum Plus sponsor.
All right, hello everyone. My name is Jackson Buss, and I'm a research scientist at NEB. And today I'm going to talk to you about miniaturizing cell-free protein synthesis to enable high-throughput workflows. Uh-oh, wrong one. Okay. So here I'd like to acknowledge the 3 different sections of research that we wanted to highlight for the ABRF community here: protein research, glycan analysis, and genomics research. As many of you are aware, NEB has a very extensive catalog. We have a lot of diverse tools and capabilities, and we'd love to share this with you all. So please come visit our booth and also come check us out at the reception to follow. Of particular note is our mass spec facility. A lot of people are unaware that we've invested heavily in mass spec in the recent years, and so please come ask us about it. I'd also like to draw your attention to several of my colleagues will be presenting posters, and as you can tell from their titles, NEB's involvement at ABRF has largely been dominated by NEB Next over the last however long. But today we wanted to do something a little different. We wanted to spice it up and talk to you about how you can use miniature cell-free protein synthesis reactions to enable your high-throughput workflows. We envision this coupled with the Boone and DNA synthesis to really enable labs to interrogate proteins of interest at scale. All right, so here's an intro to what I do every day. I'm involved in high-throughput protein engineering, where we design and build constructs and then test them in some high-throughput fashion, whether it be fluorescence or CE-based or handed off to our mass spec colleagues. And what we found over the years is that the real bottleneck is the expression of our target. And again, that's because with the recent drop in DNA synthesis costs and our dramatic increase in large sequence or large fragment assembly methods like Golden Gate assembly. The expression is the hard part. So we'd like to promote our two major products, NEB Express, which is a lysate-based product, and Pure Express, which is a reconstituted mixture. So let me paint a little context here, establish a little context. So most of you are probably familiar with traditional protein expression, where you take a plasmid encoding some sort of recombinant protein, transform it into bacteria, grow it up into a large volume, high density, and then purify it. And as anyone who's done this before, I can tell you it takes a lot of time, it's difficult to reproduce, and the throughput is limited, right? So we thought there's got to be a better way, right? So we moved to cell-free protein synthesis, which uses the same components, the same essential components to achieve coupled transcription translation, and these would be, tRNA, ribosomes, RNTPs, and whatnot. And all the user needs to provide is some sort of template in the form of a DNA, RNA, circular, linear, however you have it. And it's great. It go from template to protein in a matter of hours. And we have a lot of information, a lot of published articles and app notes on the diverse products you can use to generate, that you can generate using this. So we can make active enzymes, high binding antibodies, fluorescent proteins, or even multiple proteins at once to assess either protein-protein interaction or complex activity. So the advantages of cell-free protein synthesis are many. First of all, it's extremely fast. After you make the mixture, you can generate protein analytical amounts in as little as 2 hours. It's extremely reproducible as it's a defined mixture, and its throughput is unlimited essentially. It's a liquid, and it's compatible with all sort of liquid handlers, so as big as you can get. But in reality, that's not quite true, right? In reality, the scope is really limited by how much you're willing to pay for our reagents, and so we thought we wanted to figure out how to increase the accessible throughput of these products to our customers. So this is where we brought in the miniaturization, and we came across the same issues just in terms of efficiency. We're using 10 microliters of something, and we only use 1, so we're wasted 9. So miniaturization really was the key to run lean. So the first thing we wanted to test was using our favorite automated liquid handler, the Echo 525. We wanted to see if we could automate the transfer and the assembly of these mixtures. So we compared that to our typical way we do it, by hand and manual pipetting. And we used 2.5 microliters, which is about as small volume as we could manually pipette, considering it takes about a half microliter of DNA. So this is using, again, 2.5 microliters, about 20-fold reduction. And so shown here is data for manual on the left, automated on the right, and 10x biological samples. And you can see, A, the reproducibility is quite nice, and B, that we were able to automate this. I'd just like to point out the CVs we got from this were around 10%, and that was true for all the data shown here, as well as in the app note, which I encourage you all to check out. It goes a lot further into detail. All right, so the next thing we want to know is how well do these miniature reactions perform? So we performed an automated DNA titration of these synthesis reactions. And so what you're looking at here is at low DNA input, or low template, there's low product, so fluorescent protein. And as we increase that, you can see it sort of tops off, saturates, and so that is the maximum yield we can get from the reaction. And what's amazing about this is the sensitivity. So this is a miniature 20-fold reduced reaction, and yet it still remains highly, highly sensitive to the input. It's about 2 nanomolar input, which if you do the math, it's it's about 1 nanogram or something like that. And again, this is producing about 0.5 mg per mil, so analytical amounts. So the next thing we want to do, now that we know it works so well, we wanted to see how low we could go. So that was 20-fold reduction. Here we're looking at, so 2.5 is 20-fold, we're looking at 1 microliter, and we're looking at half microliter. And so you can see on the left, we're doing DNA titrations within the linear range of NEB Express. And you can see that all 3 reaction volumes have a similar trend. They increase with increased amounts of DNA input. And if we plot the highlighted data and show that on the right, so plot the data according to reaction volume, you can see that the yield is linear. And this is tremendous because it's telling us we're not losing any sort of activity as we miniaturize. So we can reliably miniaturize reactions and consistently express the same amount of protein, which is about 0.5 mg per mil. And again, this is a 100-fold reduction. So now with what you would get for a standard reaction, you can do a whole 96-well plate. And so this is tremendous. You can do this for, again, it's quick, 2 hours. You can get a whole plate for one reaction, and it really opens the gates of what you can do. So 50 microliters, so miniature cell-free protein synthesis, let me just nail this home, right? Miniature cell-free protein synthesis enables more for less. You can get more reactions, they're more reproducible, and you can do so in less time. And that's it, I came under. So thank you for your time. I guess I can take questions.
Thank you, Jackson, and thank you, NEB, for your continued and ample support of this meeting. Next, I'm going to ask Tricia Rogers up to invite our first keynote speaker.
Good evening, everyone. It is my great honor to introduce our first keynote speaker of the conference, Beth Simony. We are actually fellow Brodies, and yes, we call ourselves Brodies, so you can laugh at that. It's kind of a funny nickname. But Beth leads her own lab within the imaging platform at Broad, which started in 2021, after being a postdoc for 5 years in Ann Carpenter's group. She holds a PhD in biochemistry and molecular biology from the Blackburn Lab at the University of California, San Francisco, and a BA in biochemistry and molecular biology from Boston University. Please, let's introduce Beth to the stage, which she's already here, so sorry. She jumped the gun. Welcome, Beth.
All right. Okay, mic's working. Thank you so much for having me. It's my first time at ABRF in a few years, I think since South Carolina, so I'm super excited to be here. I am keenly aware that I am what stands between you and the reception. And the other thing is that I've been told my whole life I talk fast. It's probably not going to get fixed today. So there is a QR code for all of the slides. It's on just about all of the slides. If you want to go look at stuff later, there's even some extra resources for some things that I didn't have time to talk about. So if you missed something, the slides are there. So just going to start with why images. And I started here because I sort of had a moment of panic when I realized that, oh my gosh, I have to give a talk where not everyone cares about images. Biological images can be informative. They're also pretty. And I'm sure over the next 3 days, and especially when you go over into the exhibit hall, you're going to hear a ton about amazing techniques that you can do to do lots of kinds of omics at scale. But in terms of the ability to watch molecules interact, watch cells change over time, and to do this really at scale, microscopy still has significant advantages. We do have a major disadvantage, though, that our history is mostly qualitative. I'm very glad I wasn't doing science at this time, because this is not what my drawings look like. But for about the first 400 years of microscopy, representative image shown was enough and was how we communicated our data. So when we now move into this new quantitative era of science, we're a little bit behind. You know, for single-cell RNA-seq, nobody's ever looked at single-cell RNA-seq without a computer. Whereas for microscopy, we have all of this baggage we're still trying to unpack. I'm going to tell you a tiny bit of backstory about myself, not because I think that my story is terribly special or unique, but rather because I think that it isn't. I did my undergrad degree about a mile northwest of here at Boston University, doing lovely confocal microscopy in the imaging core at BU. And everything was about representative image shown. And when it was time to write the paper, I was told to just print everything out and stick them on the floor and then, group them and then see what it looks like. And that was how we came up with our conclusions. So, we did a little bit of quantification, we did some line profiles, but, it's not really that quantitative. I then went and moved 3,000 miles west to UCSF, where I wanted to study telomeres. And these are telomeres in red with DNA damage spots in green, and I was told just count how many colocalized things you have and call it a day and move on. But I wanted to know if the spots that had DNA damage were different than the ones that didn't, and what were the molecular makeup of these different spots. And each cell has hundreds of telomeres, and I wanted to look at hundreds of cells, and so crap, I needed to learn to code. Ultimately, I'm very grateful that I did, because that is what led me to the Broad and to the imaging platform. And the imaging platform is an amazing place. Our senior director, Ann Carpenter, as well as the other group leader, Shantanu Singh, are two of the nicest, smartest humans that you'll ever come across. And our goal is to write software and to write analytical tools that turn images into answers. And so a lot of what I'm going to talk about in the sort of the first part of my talk today is stuff that is really my lab is involved in, but is really led by the Carpenter Singh lab. So I'm really presenting the work of the whole platform here. So first, I'm going to tell you some of the cool new science and the cool new results about how software and data science are advancing high-content imaging. And then I'm going to tell you a little bit about deep learning and how we're working to try to make it more accessible. And then in the last part of the talk, I'm going to talk about how we can collaborate our way to better science, which is hopefully in fitting with the meeting theme. So it turns out that even when you're thinking about doing high content screening, this is about 10-year-old result, but I'm not sure that if you did it again today, that it would have changed very much. Most papers that call themselves high content only use 1 or 2 measurements of the cells or whatever objects it is that the biologist is studying. And we really think that's a shame, because digital images are ultimately just arrays. These are just quantitative numbers, and that's true of this little dot, and that's true of the camera on your smartphone. And when we talk about a microscope having a 1 megapixel or 4 megapixel camera, what we sometimes forget is that that means that there's 1 million quantitative pieces of information, or 4 million quantitative pieces of information every single time we snap that camera. And that might be across multiple stains. There might be multiple kinds of objects, multiple time points, multiple z-planes. And so there's a tremendous amount of information there. And so reporting 1 or 2 measurements just really feels like we're leaving a lot on the table still unused. And so that idea is essentially what created Cell Profiler. Ann Carpenter and Ray Jones wrote the first version 20 years ago this year. We're going to maybe have a little birthday party. We're trying to figure that out still. And so in software years, this is ancient. But what I think has really helped Cell Profiler, and I mostly want to show this slide because I want to show, we have hundreds of contributors over that time period. These are sort of just some of the major ones. If you look at the sort of outlines, we have people who are outlined in blue who are professional computer scientists, and we have people who are outlined in green whose initial training, including mine, is as biologists. And so I think the interplay of having really good computer science and really good software design, but also a lot of input from people who actually understand how these experiments work, has really sort of helped the software move on. This isn't a Cell Profiler talk, so I'm not going to spend too much time talking to you about it, but I think there are a couple of things that sort of resonate with the kinds of themes that I'm going to talk about later, so I just want to mention them. The magic in Cell Profiler is that it is ultimately at its heart an image analysis workflow tool. Those of you who are here from sort of bioinformatics cores, you understand the value of creating these sorts of workflows where we have multiple operations, we can maybe string them together in different orders and configure each one of them, but ultimately this is a workflow, and a workflow that is shareable and is reproducible. We can configure each step. We call each one of these steps in this workflow a module. Each module has help text associated with that, and I'm going to talk about that in just a second. And there are 2 modes. There's a test mode where you can just go through and sort of experiment with, all right, if I change this setting, if I add this, if I subtract that, how does my output change? And then once you've done that, you can set where your output's going to go, and you can analyze however many images you have, whether that's 3 or whether that's 3 million. Sorry, I'm going to stop hitting that. There are about 95 different modules in Cell Profiler in terms of things that you can do, and that's almost 1,000 settings the last time I counted it. And so documentation becomes really critical. I've been using Cell Profiler for 14 years now. I still forget that there's stuff that it can do. I find a setting and I'm like, what? We do this? So documentation is, depending on how you count, 25 to 30% of the lines of code. And we think that's really critical for helping adoption, because anytime you have something that has 1,000 possible options, you need to write it down, why you're doing what you're doing. Just a couple more things about Cell Profiler that, again, I think are valuable in terms of thinking about workflows. The ability to add, subtract, reorder the steps in your workflow, the ability to sort of turn them on and off and see how they affect what you're going to get, and to check how individual configurations affect not maybe just one step, but maybe a whole series of steps. These are the sorts of things that allow a user to sort of really gain confidence, and especially the fact that it's going to work not just for one module, but for all the image sets that they have, or all of the ones that they're willing to sort of sit down and test. So what does this get us beyond the ability to analyze lots of images in the same way? And that's not nothing. That is a huge thing, actually. But in terms of new advancements, what this really Cell Profiler helps with is making lots of measurements. And none of these measurements are sort of particularly special. They're things you can find in lots of tools. But it turns out just having a lot of them really helps. And a lot of them are some of the things that if you're probably thinking about in terms of what a microscope measurement is, things like the brightness of individual fluorescent channels, you know, the mean, the median, the standard deviation, all those fun things. If that's not quantitative enough for you, you can say, all right, I want to divide the cell into concentric bins, and then say how much of my fluorescent signal is in each bin, and then is it equally distributed across the whole bin. You can measure objects for their size, their shape. Again, these are sort of the things that human brains think about when we think about measuring objects. But it turns out that in addition to all of these measurements that humans think about, there's a lot of information that's hidden in images that isn't really accessible to the human mind, the human brain. My flow cytometer people in the audience, this is imaging flow cytometer data, where we have cells in different cell cycle phases. In the interest of time, I'm not going to go through the exercise where I ask for a volunteer to name the cell cycle phases and no one responds. But you might have guessed that this is a sort of ordered series, but for the first several, it's really hard as a human to sort of figure out what this is. But a computer has no problem with this. Based only on bright field features, you can get pretty high accuracy in terms of cell cycle phase. And even the mistakes that it makes. Sometimes it confuses S for G1 or G2, but the conversion from G1 to S to G2 aren't perfectly switch-like processes. So some of this is just about the fact that we are forcing things to be discrete and be in bins that aren't discrete and aren't in bins. And so this is just a pretty spinny movie showing that if we look in feature space, we see cells clustering by their cell cycle phase. So there are measurements here that matter, but do not work with human eyes and brains. And those can be things like Zernike polynomials, which is essentially how much does a particular object shape match one of these weird circles. And this makes, if I tell you that a cell has a high Zernike 3,3, you're going to look at me like I'm insane. But it turns out that these polynomials, when you add them all up, can actually give you reasonable shape description, at least much better than things like perimeter and orientation. And so measuring them, even though they don't make sense to human brains, is valuable. Likewise, things like texture measures. Some texture measures are human intuitable, but some of them are very, very much not. And all of this goes to the idea of image-based or morphological profiling. The idea that if we treat images like any other piece of quantitative data, we treat it exactly like all these other kinds of sort of experiments at scale that have been coming out for the last 20-something years. Can we learn interesting downstream analysis? Can we map relationships? The one thing I do want to point out is we say here high throughput staining and imaging, and this can be hundreds of multi-well plates, but this can also be one 96-well plate hand pipetted by a grad student. I can show some data from a paper later where that's exactly what happened. This can scale quite a lot, and we have amazing facilities at Broad who do that, but it doesn't need to be. It can be relatively small. And so this is one of my favorite papers from the early part of the field. So here we have a nuclear stain and two cytoskeleton stains, actin and tubulin. And the experiment here is, okay, we have drugs from all of these known mechanism of action classes. We know how these drugs work. Can we measure these three stains? And can we then say which drugs belong together in the same mechanism of action class? Can we group like with like? And 67% accuracy is probably not that exciting to you until I tell you that that's just the nucleus. Protein synthesis is getting perfectly called. That's not happening in the nucleus. When we include all the compartments, we get to 94% accuracy. And even the mistakes, again, are sort of making sense. DNA damage and DNA replication are incredibly intertwined. And so the mistakes here are probably just as much about our necessity to stick things into categories than they are actually mistakes. So with results like this in hand, this is where the Carpenter and Schreiber labs at the Broad started to develop what we call the cell painting assay. And the idea was if we take standard 4-color plate reader microscope that you can find in just about any imaging core in the country, how many dyes can we possibly cram into this thing so that we can image as many organelles as possible, so we can get as many measurements as possible to do these sorts of high throughput analysis and do this sort of unsupervised clustering. And so it turns out that we can cram about 8 organelles into a standard 4-color microscope. And when I first came to Broad and this was still sort of proof of concept, I was like, there's no way that this works. But it does. I've seen the light, I've drunk the Kool-Aid. If you're not a RAS pathway biologist, don't worry, I'm not either, but hopefully what you can get from this. So these are clusterings of gene overexpression based on cell painting. And the idea, what you can see is that the wild types, when we have duplicates, all cluster with each other, that's very nice. And then the next cluster over are the mutant versions of these genes that cause cancer. So cell painting is subtle enough to tell not only that these genes are kind of like the non-mutant versions, but they're a little bit different. And so Mohammed Roban, who was then a postdoc who now has his own lab, did a lot of time looking at clusters like this and seeing pathways between pathway connections that make sense and were literature supported. But there was at least one between NF-kappa B and Hippo that wasn't. And so we're a totally computational lab. We just have laptops, but we have amazing facilities at Broad who can put together experiments for us. And what they did was show that if you make a Hippo reporter gene and you overexpress NF-kappa B, Hippo reporter goes way down. And so purely from the fact that these cell painting profiles looked opposite-y, we were able to discover novel pathway biology. And so this is when I sort of drank the Kool-Aid. I was like, okay, wow, this actually works. And in the 6 years since this paper, we've started working on a lot of applications of this and how we can sort of push this idea further. Drug resistance. Can we separate cells that are sensitive to a particular drug to cells that are resistant to a particular drug? If you can do this, then all of a sudden you can start testing patient samples, and so this becomes quite valuable. It's not perfect yet, and this is the one where I said the grad students pipetted all of these plates by hand. So this is what it looks like when it's hand pipetted. It looks even better when you automatically pipette it. Plus you have happier grad students. We don't even have to be in the right cell type. So this is work that will be pre-printed pretty soon. These are skin samples from patients with various psychiatric conditions. These are not brain biopsies. These are skin samples. But there are things in cell painting feature space we can see that differentiate some of these conditions, which means this is a biomarker. And anytime we have a biomarker, again, it's a biomarker on the wrong tissue type, but it's a heck of a lot easier to get people's skin than people's brain biopsies. And now potentially we have a screening tool. Folks who work on other omics, I promise I didn't forget about you. I know not everyone in the room cares about microscopes. So this is work where we're comparing the L1000 transcriptional readout with cell painting on 1,000 drugs across 6 dose points, and seeing that there are certain phenotypes that cell painting finds better. There are certain ones that L1000 finds better. But you can synergize, and you can often get more sensitivity and more specificity when you use both. And this is a fun preprint that just came out a couple months ago, taking iPSCs from many people and working with a bunch of labs at Broad to then do GWAS of these patients. And now we have a sort of whole GWAS matrix times a whole feature matrix with a whole bunch of cells. And then can we see where genotypes are actually driving cell phenotypes? It turns out that biology is really interconnected. And so pulling out super clean data here, not that easy. But there were a couple of cases where we found really nice, clear associations between a particular genetic variant and change in a particular morphological feature. And so not content with that, the folks went in and they actually CRISPR'd these mutations in to see, can we have causation, not just correlation? And in all 3 cases where we had this strong association, when you CRISPR in the genetic variant, you see the change that you expect in the morphological feature. Now, the one on the left and the one on the right are these things that don't make sense to human brains, but the one in the middle does. This is how symmetrically the mitochondria are arranged around the nucleus. Why is this changed in PRLR? I have no idea. Someone should go figure that out. But again, it's a biomarker. It's a thing that we can use, and we can have an idea of what experiment comes next. In addition to just sort of doing this, using the same data with multiple kinds of omics, one of the really amazing things that's been coming now is the ability to bring in tools from other omics. So Harmony was made for single-cell RNA-seq data, was not made for imaging data, and I wish I had more time to tell you about this particular experiment, but I don't. But take my word for it that this is cell painting made in 4 physical locations, and we're trying to map the same compound plate imaged in 4 places. And John Arevalo, who's in the Carpenter Singh lab, who's led this work, he used sphering, and it did okay, which is one of our standard sort of transforms. It's not that fancy. But Harmony, it turns out, gets all of the sources really nicely integrated, all of the information from different physical sites. And if you're wondering what these little islands are, it turns out these are the positive control compounds. And so we are really excited to use tools that are coming from all of these other amazing fields that are doing this amazing work with high volume data, as well as for our tools to hopefully help them too and be able to sort of pass tools back and forth. The last couple stories I want to tell you here, we can now start thinking about, again, if we have this predictive power, we can start predicting the outcomes of assays. So we had an assay that was run a lot at the Broad called the cell health assay. You would have 2 plates with 7 dyes, and you can sort of see what's going on with certain perturbations, and if it's affecting the cell health. Well, cell painting, we have 5 dyes instead of 7, but it only takes 1 plate. Can we train an regression model to predict the cell health assay and then not have to do it anymore? So 50% of the time, it works 100% of the time. It turns out that some things we can predict really well and some things not quite so much. But if you care about one of the phenotypes at the top there, across cell lines, you don't have to do this assay anymore. You can just do cell painting, which gives you a lot of other information too, and then just figure out what the cell health assay would have looked like. Juan Caicedo and Tim Becker, who were then Carpenter Lab postdocs, and Nikita Mashkov, who's now Juan's grad student in his own lab, took this idea and they put it on steroids and they brought deep learning into it because we have to have deep learning in everything, and said, can we take compound structures and gene expression data and this cell painting data, and what kinds of assays can we predict? They don't have to be imaging assays, just like, can we predict yeast stuff, can we predict fungal stuff, what can we predict? Because imaging is relatively cheap and relatively easy to scale, and we have a lot of good technology for scaling it now. So some of these assays might not be so easy to scale. And again, sort of 50% of the time, it works 100% of the time. But there were a large number of assays that you could predict. And from morphology data, which is MO here, has the most number of assays. And so as you start building up bigger and bigger data sets of cell painting data, you can start doing this first, do an in silico screen first, and then just do your very annoying, very expensive experiment with a much more enriched population of treatments. And if what you're saying is, well, cool, but I don't have a giant database of cell painting data, you do. We've been putting all of this stuff up on the Cell Painting Gallery, which we are very grateful to the AWS Registry of Open Data for letting us dump a lot of cell painting data up on the internet. It's 600 and something terabytes. About half of that is image data, and about half of that is measurements of images. So go nuts. It's CC0. You can do whatever you want with it. We'd like if you cite us, please. But so to sum up this part of the talk, workflow tools can help make image analysis more accessible, and they can help with feature extraction, which I hope I've now convinced you is actually really valuable. Because these features can stand for biomarkers, even if we don't understand them with our limited human brains. And we can combine this with other omics and use the same sorts of software and start building up these really cool workflows where we use tools that somebody else invented in a new kind of approach. And we can even predict some of our assays that are harder to scale. And I'm sure there's going to be a ton at this meeting about how to make some things scale, but for the things that don't, this approach might help. Switching gears here, I mentioned deep learning and how we have to use deep learning now in everything at the end of the last section. And it's because it's valuable. Deep learning does help with a lot of stuff, that activity prediction thing that I just mentioned, and virtual staining, feature extraction, but where it's really taken off so far in microscopy is cell segmentation. And when I say segmentation, for those of you who aren't microscopists, I mean this sort of the drawing of the line around the circle there, which is a nucleus, to say, where does one object start and the next begin? And a couple of really popular networks such as Stardust and CellPose have really revolutionized this over the last few years in microscopy imaging. The problem is, installing these things is not actually so fun. So I'm going to mention our help forum a bit later. So these are a bunch of posts from our help forum about how our plugins for CellPose and Stardust don't work. And it's not just that we're bad at our jobs. It's that this turns out that this is really hard. And particularly, the people who are the subject matter experts. So this is a survey where we asked folks a bunch of questions, but one of them was, how much of your time do you spend generating images versus doing image analysis? As well as, how comfortable are you with generating new computational skills? And it's maybe not surprising that the people who say that they spend less of their time on their laptop are less comfortable developing new computational skills, but this is ultimately a problem because the subject matter experts who really understand the biology and what the biology is supposed to do and supposed to look like maybe don't have the skills to sort of use all of these fancy new deep learning things that people like me are building. And when we think about what we're building, some of the things that people are building now are great, but because they try to do everything all at once, they're just really hard or cost prohibitive. So this is Facebook's Segment Anything. It came out a couple of months ago. Anything is maybe a bit of a stretch, but if you look in the data, or in the paper, 256 A100 GPUs for 68 hours. An A100 GPU costs $10,000. So this is about $2.5 million worth of GPUs. You don't have that, I'm presuming. I don't have that. So we need to think about approaches that don't rely on everyone having $2.5 million worth of GPUs, because we want our tools to be available to anyone. Whether you're in the global north or the global south, funding budgets are not great, and this is something that a global community wants to do. So that led us to develop a tool that is a web app called Piximi. And our tagline here is images to discovery. The website's piximi.app. You can do this on your phone or your laptop right now. With the idea being that if we have a web tool, people don't have to install anything. They just open up their browser, and anything that has a browser, so we maybe don't have to rely on everyone having up fancy GPUs. We can just rely on the actual computers that we find in lab cores and do things that biologists care about, like segmentation and measurement and classification. I've shown you a bunch of classification experiments already. To do segmentation, we want to also provide annotation, because it turns out that these things where you do a first pass at the segmentation and then you fix all the mistakes and then retrain are really powerful. If you like images and you haven't tried CellPose 2, go try CellPose 2. It's so good. But so we needed to have these 4 major components in our web app. If you go to the tool right now, this is what you have access to. Annotation works really well. Classification works really well. Segmentation, so soon, so soon. I'm going to show you some videos from the demos of segmentation, probably by the end of this month. And then measurement will be the last thing that we build out. But what this is going to allow people to do is to do image analysis, and even relatively fancy deep learning image analysis, just in their web browser. So this is what the tool looks like when you open it up, when you visit this URL. If we go into the annotation component, you can see here we've pre-added a bunch of annotations of nuclei and cell membranes. And the annotation component has things that you expect, like bounding boxes and pen tools, but we've also got some fun things like superpixels. We have this color annotation tool, which I love, because especially when you put this together with hotkeys, this makes it go so much faster. I've spent a lot of time hand circling nuclei. This is better. And so here's a segmentation demo coming soon. So here we are using a Stardust model on a histology image. The model is in the website. The user didn't have to upload anything. They just point at it. They just upload their image. They hit predict. And in the amount of time that I'm taking to tell you this, I have sped this up a little bit because it's a short talk. There are the nuclei. It's not perfect, but it does a pretty good job for taking only a couple of seconds and the user not having to do anything, just pick out the right model. For the software folks in the room, we are using existing standards. We're not building our own. We're using things like COCO, so we can find also, not just cells, but also balls and clocks and things like that, but so that we can interoperate with other tools. Here's the classifier. So this is an experiment where we have cell crops from cells that have GFP, and either they don't have any GFP, they have it in the nucleus, or they have it in the cytoplasm. And so the GFP is right now red, which is wrong. So I'm going to go through and I'm going to fix that here. And I'm going to take my color map and I'm going to apply it to all of the other cell crops. Once I've done that, I'm going to save things, because saving things is always good. All I have to do to train a neural network to separate these three groups is to go into the predict tool. And all I'm going to do, so all of these settings that are the kinds of things you might change when training a neural network are hidden by default. All that's really important is that fit classifier thing in the top right. If you want to change these things, we have lots of documentation about what they do and how to change them, but you don't have to, and you can also just kind of treat them as knobs and not really sort of understand what they do and just sort of, mess around until you get something that works. So the first network that we trained is kind of okay. It's not great. So what I can do is I can say there are 600 cells with no labels, guess what the label should have been, and tell me what you think sort of nuclear or cytoplasmic GFP looks like, and I'll go through and tell you all the places where you're wrong. And I go through and train that again, and I've actually cropped this movie in the interest of time, because again, cocktails. In the original movie, I trained this thing about 5 or 6 times, plus a couple of rounds of fixing errors. It took 7.5 minutes on my MacBook. And we end up with something that's really nice at the end. Again, 7.5 minutes just on my web browser, and we can save out the classifier. And again, neural network folks in the room, this is just a TensorFlow model. You can take it from TensorFlow.js to any other TensorFlow with a super fast converter. Here it is on my cell phone. So this takes a little bit longer on my cell phone than it does on my MacBook, but it only takes 8 minutes on my cell phone to train a neural network on my cell phone. So we're really hopeful about these sorts of approaches in terms of getting these tools into people's hands without them having to have $2.5 million worth of GPUs. So in the last few minutes, and I promise I am trying to wrap up here, how can we collaborate our way to better science? So I've talked about a bunch of really cool approaches, and at least I think they're cool. I hope you think they're cool too. But to actually pull off experiments like this, there's a lot that you need to be able to do and you need to know. Some of it comes from the people in this room, the people who actually understand how these experiments work. They understand the sorts of caveats that might come up. Like, what are all of the things that can go wrong? If you don't build that into the software, it turns out it's really a problem. And so that's one thing when we train people in these tools that we always tell them. The reason they have a ton of flexibility is because we don't know your science. You know your science. You tell us about your science, and we'll tell you the right sort of knobs to turn. But we can't build this stuff in a vacuum. But just as much now, we need the exact same set of things on the analysis side too. We need this expertise. And we also need to make it so that people feel like this is something they can do. So this is percent change of the number of PubMed, I guess not percent, but the amount of change in PubMed search results for AI and deep learning and all of these cool things for 2022 to 2020. But if we ask our users in that survey that I mentioned before how interested they are in learning more about deep learning, does anyone want to guess? Is it 50%? Is it 100%? Did anyone guess negative 10%? Because we're not doing a good job of making it clear that these things are for everybody, that these things can be used by everybody. And we haven't made a good job of making that true, but we certainly haven't gotten the message out in the places where it is true. And the fact of the matter is, in terms of expertise, we have a lot more people who come from the wet lab than who come from the dry lab. This is the NSF survey of earned doctorates. Not that a doctorate is needed for great science at all, but just as sort of a mark of the amount of trainees we are making over time. And in addition to just expertise, this is also an equity issue. I'm not saying biology is great in terms of gender parity or racial and ethnic diversity, but it's better than computer science. So if we want to build an equitable force of computational biologists, pulling from biology is going to get us there faster. But we know that these computational tools are what people want. When we asked at SLAS last year, folks in the high content imaging screening group, what are you hoping for? They want ML-driven analysis. They want Skynet that's going to do all of their work for them. I want Skynet that's going to do all my work for me, so I don't blame them. But when we ask them in a completely different non-data analysis related question, what do you wish the people that you were hiring these days knew? They wish they knew Python. So there is a big gap here, and we can't deny that there is a gap here that we need to try and cross. I'm going to go through this part really fast because I don't know how many tool makers are in the room, and probably some of you I've already heard this in a bunch of places, and I'm probably preaching to the choir. But in-app documentation, I don't know if it needs to be 30% of your code base, but it needs to be enough of your code base that your users feel like they know what they're doing. And bring in users at this stage. So this is the cell profiler. These are counts of changes to the code in a period where we were specifically rewriting the documentation. And again, going back to that green-blue software engineer versus biologist, most of the changes here are being driven by the biologists because they are the ones who know how biologists talk about things. When we survey people who say that they spend most of their time making data, what tool creators should do, they'd really just like some videos. They really would just like us to make some videos. And that really surprised me. We've seen this now 2 successive surveys because I am computationally relatively high skill, and people who are relatively high skill like written stuff, but people who are computationally rate themselves as low skill would really just like us to make them some videos. They would also like us to hold some office hours, so think about doing that. And centralize our sources for information. So in image analysis, this takes the form of image.sc. We started this about 4 or 5 years ago. We're up to 55 official partners, so not all, but most of the open source image analysis software tools, we have one central forum where you can upload an image and you can upload a question and say, I don't know what I'm doing, help please. And you have 50 tools where you have an expert from those tools to say, this is how you do it in my tool. I don't think we're quite there yet in terms of friendliness, but we're working towards it. There is a sister forum for sample prep and microscopy. It's called the Micro Forum, so check things out there too. Documentation. And I do just want to finish up by saying there is already a lot of good stuff out there, and I hope no one has been sitting here the last 5 minutes and being like, well, I wrote this great tutorial, and you're not talking about that at all. I think containerization is going to be one of the main ways we go forward. So a container, things like Docker and Singularity, is an operating system in a box. It's not technically quite right, but work with me here. That's close to right. The idea being that you can take any software, any piece of data, and stick it in a container, and then all the user needs to have is a tool that runs containers. And then they can use any tool without having to install anything else. Biocontainers has made 1,000 of these already. So if you're going to put time in learning how to do a particular thing, learn how to use containerized workflows. And developers make containerized workflows. We've done some stuff for running this on the cloud, but I'm going to just skip through that. Things like Jupyter or Galaxy as well, so that you can make reproducible analyses. These can involve containers, though they don't have to. It's a lot easier if you do, though. And so the more we sort of wrap these tools and make them easy to access, the better we're going to do. And there are a lot of organizations that are pushing hard on this, and I swear I'm almost done. So ABRF, I hear, is this cool place that thinks about this and thinks about driving collaborative science. AI for Life is a new organization whose logo I completely ripped off earlier. Where they're doing open calls for helping researchers. They're making a zoo of models that if someone trains a deep learning model, they can just upload it. NewBias was the European Union of Bioimage Analysis, but we've gone global and we're getting a new name. We just have to decide what it is. But making YouTube videos and running training schools so people can come and learn this stuff, as well as just making indexes of what else is out there. And I particularly want to call out Global Bioimaging Euro Bioimaging and Bioimaging North America are the two sort of biggest and oldest that have the most stuff with them, but there are ones for every region. And they host things like exchanges of experience and have lists of core facilities. So you can find your local expert on whatever and say, hey, can I come over? And they will pay for you to go do that if it's not a local expert, if it's an expert from far away. And I particularly want to call out life science trainers, which is a sort of newer, smaller group. It's not affiliated with any major organizations, but it's a place for people who run trainings to find each other and, first of all, help us all learn how to teach better. But also, they have a huge list of trainers in various things, most of which are about statistics or computational things of some sort. So you can find your local expert, and you don't have to do everything yourself. So in this talk, the things that I've told you, accessible data science and software tools are going to help us do better science. It's going to do science that we can't do yet, and that we couldn't find things we didn't know were there. We're going to make things like web tools and containers that make it easier for us to get there and to make it easier for everyone to actually be able to do this, not just in theory, but in practice. But the most important thing, maybe, we are part of a fantastic community that loves to share, and again, that's the theme of this meeting. And there's a lot of stuff already out there, so take your time, come find us, come find me at the reception. I'm really shy, so I'm not going to introduce myself to anybody, but like, you can come up to me and introduce yourself, and I'll be really happy. If you want to find me specifically, not just at the reception, image.sc, if you remember anything about image analysis, image.sc, because that's where you learn to go learn things. That is sponsored by the Center for Open Bioimage Analysis, which is an NIH grant we have that lets us do things like community engagement. And I've talked about a lot of great work here done by our sister lab, the Carpenter Singh Lab, and my lab, work of a tremendous number of people, and some people who paid for it, and thank you guys for listening. Questions? Or was it all so clear? I'm being told to remind folks to please go to the microphone so everyone can hear, and also so people online can hear you.
There's microphones up here and here, so.
Hi, great talk. It was really a lot of fun. You said that there was a slide in which you said that basically one of the things that the PIs wanted is for their new people to know how to do Python. Mm-hmm. Can you explain why that is not sufficient? I think that was not, you do speak very fast. So
I do, I'm
I
sorry.
don't have time to go through that, but I think it would be useful for me to understand what you meant.
Yeah, so.
Not that I don't agree, I just elaborate a little.
Yeah, no, thank you. In general, there are a tremendous number of great wet lab scientists these days, and we're making more and more all the time, and the people in this room are training more and more all the time. But if you're taking someone who understands biology and then also knows how to code, again, they can help with this sort of bridge crossing thing. And so I think what they want is not just Python, but biology plus Python. But Python was just what came up in the word cloud. Great question. More questions?
Great presentation. Thank you so much. I guess I had a question for you. So I think about the downstream image analysis, the beautiful work that you're doing here. As somewhat being child, no offense there, to the acquisition parameters. What's your bit depth? All of these concepts about the data that's actually going into the image analysis. And so I love the fact that we have these universal image analysis platforms that are open source, as they should be. Yeah, sorry, maybe that was a loaded comment there. I apologize. It's my data. I want to be able to work with it. So my question is, we have a lot of beautiful resources for image analysis. Do you have any resources that you like for figuring out how we equally align our acquisition and train for appropriate acquisition so that we have the full breadth of data to analyze using these platforms?
Yes, it's incredibly important. Thank you for bringing it up. Here's the helpful resources I didn't have time to talk about slide. And the top one actually gets at exactly what you're talking about. We recently put together something that we call bioimagingguide.org, which goes along with a preprint. It is incredibly important to understand how your acquisition affects all these parameters and to have it be a cycle, to have you try an experiment, pilot it out, do the analysis, see what you can and can't measure from the way you did the experiment, and go back and forth. And so that points at a tremendous number of resources that people in this room and not beyond this room have made about the right things to do for each step. So it's sort of a thing to teach you what you need to go learn in order to do this well. But yeah, there's a tremendous number of resources, and people are now working to sort of collate these.
Question from online. Is Python significantly better than R for the work you have been describing?
I don't want to start a flame war here. Python is much better for deep learning. Most deep learning stuff happens in Python these days. I mean, something like Jupyter, in theory now, you can run notebooks that are hybrid Python and R. But ultimately, the best language to write in is the one that you can write correctly and quickly enough to actually learn the thing you want to learn. If you only ever have time to learn one language, I think Python's going to be a little bit more versatile. But I love my R friends, too.
Great talk, Beth. Also, thanks for calling out us life science trainers. I had a question about Cell Profiler being its own GUI, as well as it being in Galaxy. Why both of them? If it is a graphical user interface, why do we need Galaxy?
That's a great question, and I didn't specify enough how much that Galaxy is so many things, which mostly are not image analysis, but someone has kindly built an image analysis one. The GUI in Cell Profiler is sort of indispensable in that you can sort of tweak things and see how it affects your workflow, see how the outlines of the cells that you're making actually change. You don't want to do that in Galaxy because Galaxy isn't built to do that. But then if you have hundreds or thousands or millions of images and you don't want to run that on your laptop, which, you know, respect, then you can put it in some place like Galaxy, upload all of your data, and then put it in the Cell Profiler pipeline runner on Galaxy and have it run your millions of images so that your laptop doesn't blow up.
Do we have any more online questions? Nope.
No.
Any more?
We have one more
Oh,
question.
we have one more
We
question
have
over here.
both sides.
Let's take one on the right, and then we'll go to the left.
I love Cell Profiler. I love using it for my job. I
Thanks.
also really love using QPath because it can handle those like massive whole slide images.
Love QPath.
And the one thing that always breaks my heart about Cell Profiler is when I try to put in a really large image in X and Y and it says, no, thank you. So I was wondering if there were any plans aside from having to manually break your larger images into tiles, which can be kind of a pain sometimes too, to maybe try and like merge and marry those two profiles together.
Absolutely. Cell Profiler, like I said, is 20 years old, and not so much with the slide scanners 20 years ago. So there are a lot of inbuilt assumptions that assume your images kind of fit in memory. Cell Profiler 5 we're working on right now is about solving that exact problem, because we know it's a problem. Awesome. Thank you.
Hi, thanks. A wonderful talk. And I might be one of those people who might be looking for videos, so my question might reflect that as well. So my question was, if somebody is looking to use this tool to look across data that comes from different labs, what are the criteria that you need to think about? Because when I think of deep learning, what I'm thinking of is learning across large amounts of data, which may not necessarily be generated in one lab.
Yeah, no, that's a really good question. It's a really hard question that I can't fully answer for you. At minimum, it's something where you can normalize to a negative control. If you've got that, is really nice. As long as with neural networks, a lot of data can be relative. Like I showed in that Piximi thing where we were training a neural network with like a couple hundred annotated cells. As the networks get better, you don't need to necessarily have a lot of data from a lot of sites. But anytime you're training a neural network or regular non-deep machine learning, having a training set that represents the diversity of the data you expect is critical. Can't tell you how many folks come and they've trained it on only their most beautiful data, and then they're like, it doesn't work on my crap data. And I'm like, well, yeah, because you didn't tell it crap data exists. If you told it crap data exists, it would try to fix it. But it's not a complete answer to your question, and I'm sorry, I wish I had a better one.
Well, I think that wraps up our first keynote. Now we all get to go to the reception and have some of those drinks. So let's give another round of applause to that.